Proteins can be described by 4 different levels of structure. Primary structure is the exact sequence of amino acid units. Secondary structure is the regular conformations of segments of proteins when they fold. Tertiary structure is a three-dimensional structure of entire polypeptide chains. Quaternary structure is only present in those proteins that are composed of 2 or more peptide chains. Quaternary structure describes the arrangement of individual chains of amino acids or peptides relative to each other. Let's look at in a little more detail at each of these different types of structure. Primary structure can be represented by bonded 3-letter codes of amino acid units. In this example, we see that lutein is on the N-terminal end and histidine is on the C-terminal end of this peptide chain. In between each 3-letter code is a peptide bond. This figure represents the primary structure of insulin. Although you don't see the peptide bonds, you can identify the individual amino acids connected and the order or sequence in which they're connected. You should also note that in this primary structure, you see that different chains are connected by the disulfide bridges or disulfide bonds that we mentioned in a previous video. We've already described that secondary structure describes the regular conformations of portions or segments of proteins when they fold. These folds are stabilized by hydrogen bonding between the hydrogen of an amide group and the oxygen of a carbonyl group for different amino acid units. The 2 most significant types of secondary structure are the alpha helix and the beta pleated sheet. In the alpha helix, the R groups or side chains protrude outward, and there's hydrogen bonding between amino acid units that are about 4 amino acid units apart. An example of proteins that have alpha helix components would be proteins that comprise wool or muscles. Beta pleated sheets have polypeptide chains that are in a zigzag structure. Hydrogen bonding between neighboring chains helps keep this structure together. In beta pleated sheets, the side chain groups are typically very small, and overall, the beta pleated sheet gives the structure a very rigid form. Silk is one example of a protein that has a significant number of beta pleated sheets comprising its secondary structure. Another significant type of protein structure is tertiary structure. Tertiary structure describes the way proteins fold to maximize stability. These folds can be held together by disulfide bonds, hydrogen bonding, or hydrophobic interactions between nonpolar side chains on neighboring amino acid units. This tertiary structure is due primarily to the primary structure or the sequence of amino acids. In aqueous environments, the polar side chains will typically be on the outside of the protein, whereas nonpolar side chains will be on the interior of the protein. In this figure, you should notice that even though the overall protein tertiary structure does display some alpha helices and some beta pleated sheets, there are significant portions of the protein that have no specific secondary structure. We've already mentioned that some proteins can have more than one peptide chain in their structure. These individual chains are called subunits of the entire protein. The subunits are held together by the same types of interactions that hold together the different parts of peptide chains. Specifically, these are hydrophobic interactions, hydrogen bonding, or even some kinds of electrostatic attractions. The quaternary structure, shown here for hemoglobin, describes how the individual subunits are arranged in space. In this case, you see that we have 2 beta chains and 2 alpha chains, and inside each peptide unit is a heme group, which is sometimes called a porphyrin, and the porphyrin or heme group has at its center an iron 2+ ion. This overall structure allows multiple oxygen molecules to be bound to the heme groups, including the iron ions, so that the oxygen molecules can be carried around the body. This figure ties together the different levels of protein structure. We have primary structure, which has the sequence of the individual amino acid units. We have two different kinds of secondary structure, including the beta-pleated sheet and the alpha helix. These two types of secondary structure can be shown in the tertiary structure, which describes the entire folding of the entire peptide chain. We have an alpha helix and a beta-pleated sheet highlighted in blue. When a protein has multiple peptide chains, then the way those individual peptide chains are arranged in space is described by their quaternary structure. The functions of a protein are dependent on the particular arrangements of the various peptide chains. We can destroy or remove these functions from the proteins by destroying the organized protein structure. This is known as protein denaturization. It's accomplished by breaking the hydrogen bonds or other electrostatic interactions that keep the proteins or the peptide chains in their particular folded conformations. There are various ways of denaturing a protein. This can be done by pH changes, which disrupts the electrostatic interactions and hydrogen bonding. We can put the proteins in organic solvents, which would disrupt the hydrophobic interactions in parts of the peptide chains. We could also use heat or agitation, which would disrupt all the different kinds of protein forces. When you heat an egg, for example, as it changes from colorless to a white cloudy appearance, essentially what you're doing is you're denaturing the proteins in the egg.